the last presentation in our program. I would like to introduce Natalia Tomashevich from Federal Research Center of Biological Plant Protection, which is located in Krasnodar, with her presentation, Microbiological Plant Protection, Selection and Study of Bioagents. Yes, Natalia, are you hearing us?
Yes, I hear, and you?
And yeah, we also hear.
Thank you, dear colleagues. Good afternoon. My name is Natalia Tomashevich. I am from Krasnodar, from Federal Research Center of Biological Plant Protection. And today I want to tell you a little about the research of the Laboratory of Microbiological Plant Protection, Plant Protection. The main direction of our research is the development of technologies for the production and the use of biopreparation for plant protection. This slide shows the main steps, and we think that the research stage in the development of a biopreparation include selection of sites and source for isolation of microorganisms, isolation of microorganisms, primary screening for sites of target activity, secondary screening, comparison with the standards, different chemical and biological pesticides, the study of morphological and physiological and biochemical properties, and development of technologies for the production and the use of the final product. In our Federal Research Center of Biological Plant Protection, we have a bioresource collection, state collection of entomer, carfax, and microorganisms. In our laboratory, we have a lot of microorganisms. They are bacterial strains with fungicidal and growth-stimulating effects, strains of trichoderma, strains of entomopathogenic viruses, and so on. We have a scientific equipment, technological line for obtaining microbiological plant protection products of a new generation. This can help us to do all research. In the following, we think that the strains must have biocontrol properties, safety, and must be technological. Some of species of microorganisms must have target biological activity in vitro and in vivo, direct or indirect plant growth stimulation, no phytotoxicity, and safety for humans, warm-blooded animals, bees, and fish. And bioagents must be manufacturability. Requirements for modern biopreparation is biological efficiency, economic efficiency, optimum titer, growth ability of a producer strains on availability substrates, good solubility for agriculture, storage stability, and compatibility with chemical and biological crop protection products and different fertilizers. In the first step, we determine the antagonistic activity of the strains from our bioresource collection. And there is the first step of that describe the biofungicide development strategy using some of the studies. Now I presented on of one or two strains isolated by the scientists from our laboratory as an example. When we described antagonistic activity, we must to study that is there phytotoxicity of our strains, and if we have some phytotoxic strains, we must reject that from screening. An important step is the evaluation of the growth stimulating effect, both digitally in the germination length and weight of shoots and roots, and visually. For example, you can see as one of our strains, Bacillus valisensis BZR 336 produce root stimulation. After that, we test the biological efficiency of the selected strains in the field. For example, this is efficiency that we study in the climatic chamber on winter wheat Batька variety. When the test will excellent, we must before proceeding the development of technology for the production and the use some strains, we must ensure that there is no toxicological assessment of strains. We do this work with our colleagues. In parallel, we study the mechanism of action of the strains, and there are some mechanism distant when we can see deformation, lysis, and disintegration of Gifa Fusarium graminearum, and contact when lysis and degradation of mycelium occurred with direct contact between bacterial strains. After that, we must to see that there is compatibility of our strains with chemical and biological pesticides, growth regulators, fertilizers, adhesives recommended for use on a particular crop. It must show us that we can use our strains in the integrated pest management. And our federal center has its own stationary crop rotation, which allows us to test previously selected samples of biological products. For example, by 3 years, 213, 215, we tested our different strains, Bacillus subtilis BZR 336 and Bacillus subtilis BZR 517 on winter wheat of Batька variety in small plots, and have that and see that biological efficiency by the years was different. But we have biological efficiency, and after that we can develop laboratory and regulations and specifications. How we do this? We select the optimal nutrient media and conditions for cultivation. During these studies, we evaluate the titer of microorganisms. There are different nutrient medians and antifungal activity, also on different nutrient media. And the interesting aspect in the study of metabolites of our cultivated strains under various conditions. On this slide, we can see chromatogram and bioptogram of liquid culture based on strains depending on the composition of the nutrient media. It was potato glucose agar, optimized nutrient media, and medium of Kinga B. We had lipopeptide standard solution there, and after that we test Fusarium axisporum and see that and can see that there are surfactin, iturin, and fungicin on different variants of our tested samples. We also, with the help of our colleagues, evaluate the amount of various metabolites using HPLC. For example, we tested 4 strains of Bacillus, and this study shown that surfactin concentration, fungicin concentration, and iturin concentration were very different in our samples. When all previous stages are passed, we study of the genome and transcriptome with the help of our different colleagues, and we understand that there are very interesting details. For example, analysis of transcriptome of strain Bazer 517 revealed of 18 genes confirmed the synthesis of iturin, surfactin, fungicin, bacillomycin, and the product of UXAL gene responsible for resogen activity by these strains. And when we study the genome of our strains, at the first step, we study the genome by 6S RNA, and at the next step, we study genome in different another methods of another methods. And when all the previous stage are passed, the stage of pre-registration and registration tests begins. And only after receiving all the permits, the preparation can be used in Russian territory. Now I will briefly talk you about what are the microorganisms we study for the development of biological products. And one of the direction is selection and study of bacteria effective against toxin-forming fungi. We have just started it, but we have already determined that there are strains in our collection that reduce the amount of mycotoxins. And there is the results when we can see that Bacillus velizensis strain of liquid culture and of supernatant can reduce mycotoxins of Fusarium graminearum. Another interesting and promising direction is the selection and study of nematocidal bacteria. Here we study the influence of liquid culture, aqueous suspension, and sterile supernatant against Milledegina incognita and another nematode. The direction of polyfunctional agent is promising. For example, in apple tree, it's actively affected by codlin moth, Cydia pomonella, and different several disease. For example, Venturia niqualis, Podosfera leotricha, Alternaria alternata, and Manila fructigena, and so on. We have selected strains with insecticidal activity against large wax moth, large flower beetle, and against codlin moth. It was 5 strains. After that, we appreciate their antifungal effect, and it was found that some strains have multifunctional effects against pests and disease. For example, this strain, and this strain. And finally, we have entomopathogenic viruses in our collection, and we evaluate their effectiveness on the test objects and target objects. On the test objects, for example, it can be Galleria melanella, and target objects, it is Cydia pomonella. And when we study entomopathogenic activity, we can see that the biological efficiency of different objects are different. Our strains are protected by patients, and there are databases with the characteristics of microorganisms dependent on their target actions. There you can see our team. Here is Angela Asaturova. He is the head of our laboratory, and colleagues that study different aspects of microbiological plant protections. There is our key publications, and in conclusion, we would like to thank Michael Semyonov for the invitation on this discussion, and we are open to cooperation, and here are our contacts. Thank you for your attention.
Thank you. Wow, some sounds. Natalia, thank you so much for your great presentation. You showed us that you can do a lot of things with construction research of phytopathogens, And I think that there are some questions. Do you have maybe someone?
Yeah, very good work. Well, we have the very similar work.
Yeah.
I have one question. In the practice, what concentration of the bacillus you used to treat the plants?
1 minute. The titer of our strains are about 10 in ninth, 10 in ninth, and after that, application rate is about 2 or 3 liter on a hectare. About this, but this different for the different strains.
Yeah. Also, more question. I saw you have the very good strain control that harmful insect, to the worm. What the species? This is Bacillus thuringiensis.
We don't know now. We don't know the species, but we know that it's Bacillus, and we think that it may be thuringiensis.
Thank you.
Okay, maybe one question from me. Natalia, against what phytopathogens do you construct your strains? So which phytopathogens can be controlled using your bacillus strains?
It depends on the strains. If we talk about Bacillus valisensis BZR 336 and Bacillus valisensis 517, it's the main pathogen is Fusarium, different species of Fusarium. And also we tested our strains on the different culture, on the different crops, and on different pathogens in laboratory and field tests, but the biological efficiency was different, and depend on crops and pathogens.
Okay, yes, it's clear. And maybe some other questions? Yes, no? No? Okay, thank you so much for your presentation.